Assalamu alaikum. I am Habiba Munir and I am presenting my presentation to Professor Shahbaz. So the topic of my presentation is about nucleus. Discovery. It was discovered by Robert Brown in 1831. So presence of cell nucleus was discovered by Robert Brown in 1831. Its early discovery was undoubtedly due to its prominence in many cells, where it stands out as slightly darker than the surrounding cytoplasm. Then location. In animal cells, it generally occupies central space, while in the case of plant cells, it is pushed towards the periphery due to the presence of large vacuole. Nucleus may be irregular or spherical in shape. Then appearance. Nucleus is only visible when the cell is in non-dividing stage. It contains chromatin network and soluble sap called nucleoplasm. In dividing cells, the nucleus disappears and the chromatin material in it is replaced by the chromosomes. The hereditary material is in the form of chromosomes while controls all the activities of cell. DNA, RNA and proteins including enzymes form the chemical composition of the nucleus. Composition. Nucleus consists of nuclear membrane, nucleoli, nucleoplasm and chromosomes. Nuclear membrane. Nucleus is surrounded by nuclear membrane which separates the nucleus material from the cytoplasm. The nuclear membrane is actually a nuclear envelope composed of two membranes. The outer membrane is at places continuous with the endoplasmic reticulum, while the inner membrane encloses the nuclear content. The outer and the inner membranes are continuous at certain pores, resulting in the formation of pores, the nuclear pores. The nuclear pores allow the exchange of materials between the nucleus and cytoplasm. The number of nuclear pores is highly variable. The undifferentiated cells have numerous pores, whereas differentiated cells such as erythrocytes have only 3 or 4 pores. Each pore has definite structure which control the traffic of substances passes through them. Nucleolus. It is darkly stained body within the nucleus and is without any membranous boundary to separate it from the rest of the nuclear material. There may be one or more nuclei in the nucleus. The ribosomal RNA is synthesized and stored in the nucleolus. It is composed of two regions: the peripheral granular area composed of precursors of ribosomal subunits and the central fibril area consisting of large molecular weight RNA and rDNA. It is the nucleolus where ribosomes are assembled and then exported to the cytoplasm via nuclear pores. Chromosomes. Nucleus is often deeply stained with basic dyes because of the chromatin material. During cell division, chromatin material is converted into darkly stained thread-like structures known as chromosomes. Under a compound microscope, chromosomes appear to be made of arms and centromeres. Centromere is the place on the chromosome where spindle fibers are attached during cell division. Each chromosome consists of two identical chromatids at the beginning of the cell division, which are held together at centromere. So it was nucleus. बेसिकली न्यूक्लियस होता क्या है? न्यूक्लियस सेल के अंदर मौजूद एक ऑर्गेनेली है. इसकी अपीयरेंस के बारे में हमने पढ़ा, इसकी लोकेशन के बारे में पढ़ा, डिस्कवरी के बारे में पढ़ा और इसके स्ट्रक्चर में आ जाते हैं न्यूक्लियर मेम्ब्रेन, न्यूक्लियोलस एंड देन क्रोमोसोम्स. ये इसकी डायग्राम है. इसमें न्यूक्लियर पोर्स को दिखाया गया है, देन न्यूक्लियोलस को दिखाया गया है, न्यूक्लियोप्लाज्म को दिखाया गया, क्रोमेटिन नेटवर्क को दिखाया गया, न्यूक्लियर इन्वेलप को, और ये सेल के अंदर मौजूद न्यूक्लियस है. अब